Thank you so much for having me. Can everybody hear me? We're good? So it's super exciting to be here and see so many people excited about machine learning at Rice. Very exciting times. So today I'm going to talk about learning networks from diverse data. And so actually in the first two talks, it was great. We heard the different types of diverse data that are coming out of the energy industry. But diverse data is in so many sectors these days. Just think about national security type data, where for the same subject, you probably have geographic information, call logs, maybe their internet browsing history, their social media postings, and so forth. This is all different types of data, and a fundamental challenge when we've got diverse data is how do we integrate all of this data and put it together and make sense of all of this data in one model via one machine learning method. And again, this type of data is in lots of different technologies. I'm going to specifically talk today about biomedical technologies. To just kind of get on the same playing field for what I mean by this type of diverse data, I specifically call this type of data mixed multimodal data, where the data we have for the same set of subjects, observations, samples, the same geographic location for reservoirs, for example, we have multiple sets of features or different sources of data that was measured on this same set of subjects here. Okay, so we call this mixed or heterogeneous data sources. And this is type of multi-view data or multimodal data that has many different types, and we want to put all of this data together. And I define statistical data integration as what we would like to do is to use probabilistic models to somehow jointly model all these multiple types of data on the same set of subjects. So we have, for example, in machine learning and in statistics, we have great models to model one chunk of data in this kind of data matrix form, or this chunk of data, for example. But how do we have a model that can encompass all of this data, put it all together, and allow us to do inference and gain actionable insights from all of this data? So the specific motivation that I'm going to talk about today comes from cancer genomics. But again, keep in mind, diverse data is really everywhere. And the framework I'm going to talk about is very general. So the Cancer Genome Atlas is a particular motivation here. This was a huge NIH-funded project that studied 33 different types of cancers. Over 11,000 patients and tumor samples were taken. And the goal was for every tumor sample taken to use multiple genomics technologies to profile and get a complete genetic snapshot of those tumor samples. So this is 2.5 petabytes worth of data. Pretty big data out there in the genomic sector. And there's many different kind of views or multi-modes of this data. So for example, the types of genomic technologies available don't give you, with just one technology, you can't have a complete snapshot of what's going on. Just like in the energy industry, there was that data gap. There was multiple different sources of data that gave you different snapshots. We have the same situation going on in genomics here. So we've got sequence changes here. These are individual points in the DNA that can change, or copy numbers that can change. We've got genes being turned off or on, measures of this with gene expression, and epigenetics here. And all of these are just one snapshot of what's really going on in tumor cells. The reason it's so important to study these is because when someone develops cancer and a cell becomes a tumorous cell, it's not just one thing that typically goes wrong. It's never just one thing. It's literally hundreds of very complex interactions that go on that make a cell a tumor cell and make it proliferate and turn into a tumor. So it's really important to study all of these facets to really understand these complex interactions that cause cancer. And specifically, I'm just going to zoom in on part of these genomic technologies so we can actually visualize things on the slide. And this is, we want to study gene expression, which loosely is defined as how much a particular gene is being turned off or on in that tumorous cell. Okay? So it's a continuous measure of a gene being turned off or on. These are really important to study because every cell in our body has the same DNA sequence, yet we have different tissues. We've got lung tissue, brain tissue, heart cells, and so forth. All of these cells are different because the genes, even though there's the same DNA sequence, the genes are turned on differently in different cells, okay? And so this is really important to understand which, in the tumor cells, which of those genes are being turned on that cause the tumors to grow and proliferate, for example, and which ones cause it to become a tumorous cell to begin with. And then the other part of this picture that we're seeking to study are the epigenetics. And this is a really cool, this is rather new technology that comes out of biomedical industries to measure epigenetics, because these have microRNAs and methylation loosely have the ability to turn off genes, okay? So, and these are both measures of how genes can be turned off. And what's interesting about this is, again, our DNA sequence doesn't really change too much from cell to cell, but the epigenetics can vary wildly, and these encode both our lifestyle and environmental factors that we've accumulated over our lifetime. So these can be really keys to studying cancer, okay? What we really want to know is we want to know how these guys change gene expression to really understand potential causes and thereby potential cures for cancer. But this is a really challenging problem, okay? So not only is this data big, so the data technologies that produce this are in the order of tens of thousands of variables here, hundreds of thousands of variables, and tens of thousands of variables here, measured on several thousand subjects, okay? So the data's big, but it's also of diverse types or different distributions, okay? So for example, gene expression data is typically count valued. This is a big spike at 0, and it has a long count valued tail, okay? DNA methylation data is typically bounded between 0 and 1. So here you see a big spike at 0, tails off at 1. MicroRNA is continuous, okay? So just imagine we've got data, some of which is counts, some of which is bounded continuous, and some of which looks nice and sort of Gaussian-like, right? How do we put all of this data together in the same model? If all of our data looked like this, nice bell-shaped curves, Gaussian stuff, the world, it's quite nice. We know as machine learners exactly how to model Gaussian data in a multivariate sense, but how do we model all these other types of data? And the approach that I'm going to use to help model this data is a framework of networks. So what you're looking at here looks like just a massive hairball of a network, right? And this is actually over 1 million biomarkers from glioblastoma, and this is an estimated network of all the connections between millions of different genomic biomarkers in glioblastoma. Okay, so this is what we're trying to do. We're trying on a holistic scale to put together all these different types of genomic biomarkers in the same model, and then to somehow make inferences about what is a potential causal factor for cancer. So there's a long history of using networks to understand genomics data. Okay, starting with the microarray technology, it was in the late 90s that this was invented. This is now kind of old technology, but you can think of our data matrix looking something like this. We've got subjects here. We've got genes here. This is tens of thousands of genes, and each little cell is a measure of that gene being turned off or on. Okay, so green means that gene is turned on strongly. Red means that gene is turned off. Okay, and from this data, the goal is to fit some type of multivariate distribution. Okay, so a probabilistic statistical distribution to this data that models interactions between how the genes work together and basically how the genes are talking to each other in the cell. You can kind of think of it like that. And the interactions or the dependencies between these genes, we can model as links between two genes in this network model. So here, each node of the network is an individual gene, and each link between two genes denotes some type of dependency or between those two genes. So this is what we're trying to do. And people have done this a lot for this kind of older technology, these microarrays that look nice and Gaussian. And this is particularly modeled with the Gaussian graphical model. There's been like tens of thousands of papers on Gaussian graphical models that have studied just this, right? This is a well-studied thing. The reason this is so important biologically is because this has saved, this type of using high throughput data to model these networks has saved scientists tons of time and money. Because before they had this access to high throughput biomedical technologies, it was kind of guesswork, educated guesswork, and they would perform tons of experiments to see which genes are talking to each other. Now, we can use high throughput technologies to guide the scientist and say, oh no, these two guys are the main players here in this particular network, and they seem to talk to each other. And we can then guide the scientist and say, no, these are the two genes that you need to test in this network. So this is how we're using these types of models to guide science. So the objectives here are, first, we want to develop a family of graphical models, and this is just a probabilistic distribution, to model this type of diverse data, this multimodal data, and also develop learning techniques to estimate these graphs. And the main reason why we're using these types of graphs, sometimes these are called Markov networks or Markov random fields, is because they are probabilistic models. And I'm a statistician by training. We really like probabilistic models because they can give us, we can study the uncertainty in our network estimates, and we can also provide statistical guarantees on how well we're going to do. And that's very important when you're communicating with scientists where they're going to be conducting experiments following up based on your results. OK, so what has been done as far as networks for diverse types of data? What's out there in the literature? So I mentioned this Gaussian graphical model. This has been very well studied. There's also the Ising, some people pronounce it Ising model, originally from the physics literature. And this models binary valued data. But until several years ago, there was a question of what do you do with count-valued data, or bounded data, or skewed data. There's so many types of data that's not just Gaussian and not just binary. What can we do with these other types of data? So the framework that myself and some collaborators developed is called graphical models from exponential families. Okay, so I'm going to skip all the math on these slides, but the key idea is pretty straightforward. If you have a count-valued univariate random variable, okay, everybody think back to your introductory stat class, which might have been some time ago for some of us in the room. So if you think back to that, if you've got a count-valued univariate random variable, which distribution would you use to model it? Poisson, yeah, why not? Negative binomial if you're like, fancy or something, right? Like we know what distributions to use for univariate random variables. So the key idea here is to leverage what we already know about modeling these in univariate land and define all conditional distributions. So this means one gene conditional on all the others, or one feature conditional on all the others, via a univariate exponential families, which we already know and love. Things like Gaussians, Bernoullis, Poissons, negative binomial, so forth. And when you do this, I'm skipping all the math, an actual joint network distribution exists. It's a multivariate distribution. It has closed form, and you can model these dependencies. So kind of cool stuff. So the end result of this is not only can we model Gaussian and binary valued random variables via networks, now we can also do this for count valued random variables, for skewed continuous or bounded continuous random variables, and so forth. So we can expand this class of networks. And this is just an example of a gene expression network estimated via Poisson graphical models from this latest technology, which produces count-valued random variables for these genes. But of course, it's not just enough to model, say, just the interactions in gene expression or just interactions in epigenetics random variables. We want to actually put them together. So how do we do this? So here, we're going to leverage, again, this assumption that we know how to univariately model each different type of feature. And we're going to say, conditional on the other features, each feature can potentially get its own distribution. Okay? So the genes conditional on everybody else might be Poisson, and maybe the microRNAs conditional on everybody else Gaussian, maybe the methylation conditional on everybody else could be Bernoulli. Okay? So everybody can have its own distribution, and I'm skipping some other assumptions, and I'm skipping a lot of math here, I promise. It's like 10 papers worth of math. And a joint distribution exists. Voila, I can do this since I'm giving a 20-minute talk and just say, joint distribution exists, right? There's a multivariate distribution here. And again, the dependencies, we can explicitly characterize these dependencies here. And the schematic of what these distributions actually look like is this right here. So we have a situation where this would be a homogeneous example of our mixed chain graphical model here. And the homogeneous example is within these blocks, we've got variables of one type, and we can actually model directed dependencies between variables of different types here, or we can mix them up within these blocks. Okay, so these are really flexible class of models. And this type of is exactly what we see here. If we look at this homogeneous network type here, this is the exact assumption we had when we wanted to model this type of regulatory genomics network. Okay? So the implication of this is that this is the first multivariate distribution that can directly parameterize dependencies between mixed types of variables. Okay? So this is really important because this is going to allow us to go far beyond just Gaussian graphical models. And now we can model all different types of diverse data in a joint probabilistic manner. So what does this mean in terms of learning the networks? So the problem is, okay, so now we've got this probabilistic model. The next problem is, how do we fit this to data? How do we learn the structure of the network from data? And this is a real challenge because those probabilistic models, there was a reason I didn't show them on the slides. They kind of look pretty nasty, right? They are computationally not tractable. They're NP-hard problems from a machine learning perspective to solve. But there's some tricks that we can factorize and learn one neighborhood at a time. And this is essentially saying for one gene, I want to find all the edges that extend out from that gene and that connect to this gene. And I can learn this one at a time. But this problem in and of itself is a challenge, okay? So imagine if we've got a response or an outcome of one particular type, say red type, maybe it's, say, a Bernoulli or a binary random variable, but we've got our covariates of all different types here. So how on earth do we actually put that together? And this is a new problem that in machine learning, surprisingly, people haven't really studied yet. Okay, so this is kind of a new problem in machine learning. And it has some big challenges associated with it that we characterize statistically. But our solution here is actually pretty simple. We actually develop a block randomized adaptive iterative lasso. And what this really does is it goes through one block of variables at a time and selects features from each data type separately. And it automatically solves these problems. It's also a fast and parallelizable algorithm. So we've taken a computationally intractable problem, and we've now developed one that can be completely parallelized and is fast and has strong statistical guarantees as well. I'm going to skip some simulations. It works well. I'm just going to leave it at that. Okay, I want to conclude with a quick case study going back to cancer genomics and how we use these types of models. So remember here, we want to understand what are the epigenetic, these are, again, the lifestyle or environmental factors that we've measured that change which genes are turned off and on in tumor cells. Okay, that's what we want to study here. And we're going to use our integrated graphical models to model these. And when we fit these to an ovarian cancer network, this is actually the result we get here. Our genes are in red, our methylation is in green, and our microRNAs are in blue. And we see already there's some definite structure we see in this network. The genes seem to talk to each other a lot. They're pretty well connected here. But if you look at this, we see some microRNAs. Remember, these are some of the epigenetic factors here that seem to be really, really strongly regulating a lot of genes. And just to show you how we're working with scientists to use these types of networks, if you look here and zoom in on one, this is a module that's surrounding one particular microRNA, miR425. It's regulating a lot of genes. See all those red genes around this? It's regulating a lot of genes. And the genes that are boxed in red are actually 6 of the top 10 most mutated genes in ovarian cancer seem to be regulated by this one guy. Okay, this miR425. So this is a big possible scientific finding. The reason this is so important is because microRNAs are very easily manipulatable as drug targets. So for example, I'm working with oncologists to experimentally test this microRNA to see if it can be manipulated in cells to possibly turn off all these other bad genes that you see here to stop tumor growth, for example. So this is the type of way that we're using these diverse networks to lead to scientific discoveries. So future work, my whole group at Rice is very interested in broadly studying diverse data, not just from biomedical applications, from lots of different applications. And yes, we're working with network models. There's actually a lot more work that we're going to be doing with network models, but we're also interested in studying things like clustering and pattern recognition for these types of diverse data as well. I should mention, if you're interested, please check out my website. There's some software available for these types of models in the R language, which is an open source software language. And this is a big collaborative project with oncologists, geneticists, bioinformaticians, computer scientists, and students at Rice. Thank you very much.
Fantastic question. Okay.
Could you say again how you overcome NP completeness?
Yeah, so the original problem is NP hard because the log, I'm going to get slightly technical, forgive me. The log partition function is NP hard, but there's a trick where actually if you ignore this and you factorize the likelihood, you can show that it factorizes down into the node neighborhoods times this log partition function. And you can actually show that if you learn just the neighborhoods, under certain settings, not in all settings, but under certain settings, it's equivalent to learning the original NP hard likelihood problem. So this is basically how we get around this. You do make some key assumptions there, but you can show that it's equivalent to solving this. Yeah.
You mentioned that lifestyle and environment affect epigenetics, right? So would you want to model them, and how can you model them?
Yeah, so the question was lifestyle and environmental factors affect epigenetics, and how do we model these? So right now, the data we have is just the measures of microRNA and DNA methylation, which again are just measures of the changes that have accumulated, but you really need another data set that is measured environmental exposures and lifestyle factors to link them. Several studies have already done so, so we know these basically encode lifestyle factors and environmental factors already. But we're interested in which ones particularly. The reason they're so important is because it's really hard to change your DNA sequence, and it's probably unethical in humans, right, to change our DNA sequence. So that would not be a viable therapeutic solution to develop drugs. But changing epigenetics is a possible solution to do this, because the epigenetics can turn off some of those genes that cause tumors to grow, for example. So this is why scientists are particularly excited about this area. Yeah.
Can you model in your network model? Can you model those factors?
We can absolutely model additional factors. Again, I was just showing you a subset of the data, but if you have diverse data sets of many types, yes, this model is very flexible. It can incorporate lots of different types of data. Yeah.
Oh, one more question.
Okay.
How do you work with the healthcare providers to source the data that you use?
Yeah, so the data that I'm using for this project, the question was about how I work with healthcare providers to source the data. The data I'm using for this project is publicly available with agreements between typically academic researchers. Because this was collected, this was a huge NIH project, the data was made publicly available. We do have other data sets that are a bit more proprietary. In general, these data sets are extremely expensive to collect. And so after they're collected, I typically have people coming to me as a machine learner, and they just beg me to work with their data because they spent so much money collecting their data. I've never had a problem. I just walk around the med center, and sometimes people just give me data, right? So I mean, I've never really had too much of a problem with that. My experience has been more that they've just been begging machine learners to come on board and help them because they've already spent so much money collecting the data.
How about one last question?
Go.
So mathematically, how you prove the convergence of your method?
I'm sorry, I didn't quite hear.
Mathematically, how you prove your convergence of your method?
How did I prove the convergence in the method? Okay, there's lots of different aspects of that. So this is using, I think the underlying principles of the theory of this are from high-dimensional statistics. So basically, they're probabilistic statements that say with very high probability for a certain sample size, certain number of features, certain number of conditions that we put on the structure of the graph, with very high probability you can recover the network structure. These are the types of statements. And the tool set we use comes from high-dimensional statistics. So this is a theory that's actually developed rather recently, over the past 5 to 10 years, has been developed.
Fantastic.
Yeah.
Let's thank Genevera again.
Thanks.